Our final speaker for today comes from the School of Biological Sciences, Dr. Matt Hall. Matt completed his PhD at the University of New South Wales in 2009 and then moved to Switzerland to work at the University of Basel, supported by the prestigious Marie Curie Fellowship. This was followed by a fellowship at Velux Foundation at the ETH in Zurich, where he looked at the evolutionary genetics of host pathogen interactions. Last year, Matt returned to Australia to take up a lectureship here at Monash and to enjoy some good coffee and some sunshine. He's currently supported by a Discovery Early Career Researcher Award from the Australian Research Council, with a project looking at how males and females experience infection differently and how this can influence the evolution of disease. Outside Monash, Matt is a keen runner and cyclist. Today, he's going to tell us about the Super Flea, a wild solution to everyday problems. Thanks, Matt.
Thanks very much. So rather than the super flea, we're talking about species of the water flea. So these little critters are really known for their morphological differences. They have this amazing ability to grow spines and shapes in response to different conditions. But what I'll talk about mostly is how we use these animals to understand the complexity of biology, the challenges that animals face and the strategies they use to cope with them. So the world is a very uncertain place. It's full of threats and challenges, and how we cope with them is often different for different individuals. So for us, we can think really broadly about the types of challenges, such as coping with climate change, how that influences agriculture. We can think more personally about where our money's coming from for our next grant round, or even what sort of subjects we're going to choose in another year. And so these challenges are always going to be different, they're always going to change, but everyone has their own sort of way of coping with them. And if we think more broadly about all the animals that we can study, there are some fundamental challenges that they all face. There is the ability to reproduce and contribute genes to the next generation. There's the decline in physiological performance and cognitive ability that happens when we grow older. And there's the threat of infectious disease, which every animal faces. And the important bit which I'll emphasise today is that individuals are rarely good at optimising all three of these biological challenges. And so the animal that I work with is the water flea. So these are transparent crustaceans. They feed on algae and they live in freshwater ponds throughout the earth, including there's a few different populations on campus. And so a typical scenario, a typical environment where they live, this is the Finnish archipelago. We were out collecting animals from here a few years ago, and they're not found in the big open expanse of water. They're found in the tiny little ponds and crevices that exist on rock platforms. And so here we have over 10 different populations of Daphnia. Some of them have existed happily for 40 years. Some of them go extinct every couple of years. But it's these small scale habitat differences which make them really interesting to study. Each one of these populations is evolving independently with some connections between them. And if we think about all the different small rock pools that are found all over the globe, there's lots of different challenges that they've come up with and lots of different strategies they've used to deal with them. And one of their big abilities to spread and colonise is that they reproduce really quickly. So this is a typical photo of a Daphnia culture, where you can have every single little dot is a different little individual. And the highest densities that have been observed are up to 3,000 animals per litre. And their ability to colonise and spread through a population is largely due to their life cycle. They reproduce sexually, so with the males and females contributing half the genetic material into a sexual egg, but they also reproduce clonally. So if we have genotypes that we've sequenced or genotypes that we know a lot about in the lab, they perpetuate in jars, and so we can culture them that way. And that also gives them the ability to spread quickly. And so for working in a lab system, these guys are pretty good. Rather than keeping them in petri dishes, we keep them in really small jars. We can have thousands at any one time in incubators. And so we use them to understand the sort of biological challenges that animals face and really why individuals are different in how they respond to these. So as an example of how well a Daphnia can cope with a biological challenge, this is a photo of a single genotype. So these animals on the left and right are genetically identical, but the one on the right has been exposed to a predator. And so a common response with these animals is to grow these really elaborate morphological changes that throw off either a predator's sort of search image, or they just make it difficult to handle on average. But the key point here is these have the same sets of genes, they've been raised in the same environmental conditions, except one was exposed to a predator scent. In terms of thinking about ageing, which is one of my main research topics, Daphnia from safe populations, which are consistent over time, tend to age much slower. So they reproduce at a later time in life because the environment's safe and it's without challenge. If they've evolved in a risky population, so small ponds, or ponds that can go boom or bust on a very fast cycle, then we see a much faster acceleration in their ageing patterns. And so the reason why this is interesting is because we can go to the different Daphnia populations and we can start to ask, are the same genes contributing to decline in ageing? Are the same processes shaping what we observe? And what are the consistencies that we see within and between them? For infectious disease is probably what they're best known for. So this complicated figure basically shows 4 different populations of Daphnia, and every bar is a different genotype. These genotypes were exposed to 4 different pathogens, ranging from a bacteria at the top, we have microsporidia in the middle, and a fungus at the bottom. And the main component to notice here is there's not one population that's completely resistant to any pathogen, and there's not one genotype that really performs equally well in any scenario. And so with working with this system, it's about trying to understand, why does this diversity exist? Why aren't animals more resistant to infectious disease, given pathogens are generally what drive populations to go extinct? And so I'll just give a really quick example of a simple study that was done from an honours student of mine. She was interested in crowding. So she took 2 Daphnia genotypes, a clone HA2 and a clone M10, named because of one's from Hungary and one's from Munich. She then exposed them to signals of either high density, such as 500 animals per litre, or low density with only a couple. And the main point to notice here is that one genotype under high conditions got smaller, one genotype got larger. If we look again in terms of reproduction and fitness in that component, we have the same thing happen. The genotype that got larger also produced more offspring, the genotype that was smaller produced less offspring. So it seems to be one's doing better and one's doing worse. If we look then at infectious disease in terms of how well a pathogen is able to evolve and grow within these hosts, we get a reversal of the pattern. So the one that was more fecund is also less resistant to a pathogen. And so it's the basic take-home message is that when considering biological challenges, individuals are rarely able to optimise which bit they're good at. And so understanding how that's evolved, what genetic component has led to be one to be better at resisting infectious disease, one to be better at reproducing, is about understanding what makes us all individual. And with that, can I answer some questions?